going through old school ID labs, old school microbiology labs, you would have test tubes, you'd have petri dishes. Well, test tubes, petri dishes take up too much space, are expensive in the long run to go and do all that. And well, it gets a little easy to start mixing up different test tubes. Was it for this sample? Was it that sample? So what's been designed? Well, you look here. This is the new method for biochemical testing you're going to find predominant in most ID labs nowadays. It's a card about the size, just a little bit bigger than a normal credit card. And it's going to have wells on it. Each of the wells is going to have a different, it's going to have different broths. It's going to have different augers that are going to be on it, in it. You go through, you take your sample, you've isolated the bacteria you think might be it, causing the infection, and you go through and put it in all the wells, and then you just put the well in the incubator, because remember you have to incubate, and you let it incubate for a little while. So instead of a big test tube, you have this little bitty well that has maybe a drop of broth, maybe a drop of agar in it. So instead of hours and maybe 12 to 24 hours for the big tube of broth to change color, the little bitty drop is going to start to change color within a few hours, 2 or 3 hours. So it goes from red to yellow, positive, green to blue, positive. So things can be done. Small little credit card size thing. It's going to have, you see here, it's going to have what, 1, 2, 3, 4, 5, 6, about 20 wells, and there's some that have more. It's a little bitty thing. Everything's bound together, so there's no mixing up. Oh shoot, which patient sample did this test tube go with? No, it's all there. It's all together. You're just going to have a visual representation. You just go through and check the boxes. For the first one, for well 2, positive. For well 3, 4, negative. For third one, positive. Fourth, 4, 5, 6, 7, 8, negative, negative, negative. And it's this pattern. You go through, you collect the pattern, you add them up, and as you see at the bottom there, it gives you a specific serial number. That specific serial number coincides with, was it Edwardsellia torta? Never heard of it, but it's a specific bacteria. Go through, you add up the positives, the negatives, the pattern, gives you a specific serial number. Staphylococcus aureus gives you another, do it again with some other sample. Add up the positive, the negatives, get the specific sequence of numbers. It's a serial number that coincides with Escherichia coli. Keep in mind that the biochemical testing is taking an unknown, and you're looking at whether or not a reaction occurs. Yes, no. If the enzyme is present, or if the reaction occurs, that means an enzyme was present. The enzyme's present, there has to be a gene present. Not all bacteria are going to have the same genes. Not all species of bacteria in the same genus. The different streptococci are going to have different genes present. The genes present in Streptococcus pyogenes are going to be different than the ones in Streptococcus epidermidis, the other Streptococcus. Staphylococcus aureus is different than Staphylococcus epidermidis. So we're looking at yeses and nos, positive, negative reaction, because positive means an enzyme's present, which means there's a gene present, which means, that bacteria has certain abilities that others won't. And it's the pattern of yeses and nos that are going to be indicative of a specific species.